A sharp mind and a bodybuilder's physique in old age almost sounds too good to be true. New research out of Chicago, however, suggests that a popular supplement in fitness circles for building muscle may also help stave off the most common form of dementia, Alzheimer's disease. Scientists at the Rush University Medical Center have discovered that beta-hydroxy-beta-methylbutyrate, HMB, may help prevent Alzheimer's progression. To be clear, HMB is not a prescription drug or steroid. It is an over-the-counter supplement widely available in sports and fitness stores. Bodybuilders often use HMB to increase their exercise-induced gains in muscle size and strength and improve overall exercise performance. HMB is generally considered safe for long-term use. There are no known side effects. Alzheimer's is a progressive brain disease that at this point has no cure. Symptoms usually first appear after age 60, slowly destroying thinking skills and memories. Alzheimer's affects as many as 6 million Americans and over 10% of people 65 and older, according to estimates. Rush researchers recently discovered that a muscle-building supplement called beta-hydroxy-beta-methylbutyrate, also called HMB, may help protect memory, reduce plaques, and ultimately help prevent the progression of Alzheimer's disease. Results from the study were published in Cell Reports. Studies in mice with Alzheimer's disease have shown that HMB successfully reduces plaques and increases factors for neuronal growth to protect learning and memory, according to neurological researchers at Rush. Understanding how the disease works is important to developing effective drugs to protect the brain and stop the progression of Alzheimer's disease. Previous studies indicate that a family of proteins known as neurotrophic factors are drastically decreased in the brains of people with Alzheimer's disease and have been found to help in survival and function of neurons, which are cells that receive and send messages from the body to the brain and vice versa. Our study found that after oral consumption, HMB enters into the brain to increase these beneficial proteins, restore neuronal connections, and improve memory and learning in mice with Alzheimer's-like pathology, such as plaques and tangles. The study findings indicate that HMB stimulates a nuclear hormone receptor called PAR within the brain that regulates the transport of fatty acids, which is key to the success of HMB as a neuroprotective supplement. If mouse results with HMB are replicated in Alzheimer's disease patients, it would open up a promising avenue of treatment of this devastating neurodegenerative disease. Key facts. 1. HMB, the over-the-counter supplement used by bodybuilders, may protect memory and slow Alzheimer's progression. 2. In mice with Alzheimer's disease, HMB has been shown to reduce plaques, enhance factors for neuronal growth, and protect learning and memory. 3. HMB stimulates a nuclear hormone receptor in the brain that is crucial to its neuroprotective role. Cognitive decline is a normal process of aging, with some cognitive abilities, such as conceptual reasoning, processing speed, and memory, being more susceptible to this gradual decline. Although non-modifiable factors, like age, race and ethnicity, gender, and genetics are involved in this cognitive decline, several lifestyle interventions, including physical activities, cognitive training, and nutritional interventions, have been shown to have a positive impact. In fact, several studies have shown that maintaining a healthy diet could be associated with slower cognitive decline and reduced risk of Alzheimer's disease. HMB supplementation preserves muscle mass and strength during aging by affecting the balance between protein synthesis and degradation. The mechanism by which HMB elicits its effects on protein synthesis in skeletal muscle is related to the activation of signaling pathways involving phosphoinositide 3 kinase, PI3K, mitogen-activated protein kinases, MAPK, extracellular signal-regulated kinases, ERK1, and mechanistic target of rapamycin. These mechanisms of action are present in the brain and are involved in brain physiology and pathology. Detailed proteomic analyses reveal that HMB serves as a ligand of peroxisome proliferator activated receptor alpha, PAR, a nuclear hormone receptor involved in fat metabolism via interaction with the Y314 residue. Despite the hydrophilic properties of HMB, it seems to be able to cross the blood-brain barrier because it was detected in brain microdialysates after an acute oral administration in rats. 15. To add to this, a recent publication showed that HMB is transported through brain endothelial cells by a proton-coupled transport system. Moreover, its efficacy has been shown in experimental models as follows. It promoted the differentiation and neurite outgrowth of neuro-2A cells. It prevented the age-related regression of dendritic trees and pyramidal neurons of rats, 18, and also, 3, it ameliorated age-related decline in cognitive performance in rats. However, these studies used either in vitro doses, which have to be extrapolated to in vivo oral doses, or a relatively high nominal dose of calcium. The most studied doses of HMB in humans with regards to body composition and muscle strength have been 1.5 to 3 grams per day in the form of calcium salt, 21. The doses in experimental animal models that showed a benefit of HMB on body composition ranged from 100 to 500 milligrams by weight of HMB. Therefore, it is not known if these lower doses will be able to influence brain functionality. The aims of this study were to reproduce the effect of HMB on brain functionality using an automated system designed for behavioral studies in rodents, using a behavioral task to assess the spatial working memory in animals to test if HMB was able to affect LTP, an electrophysiological mechanism underlying synaptic plasticity and memory storage, and to study the effective dose of HMB on brain function in adult and middle-aged rodents. The doses were selected based on the literature and body weight and body surface of animals and its relationship with human equivalent dose. Because the conversion factor for mice was twice that of rats, the doses used in both experiments were similar when calculated as HED and were approximately between 2 and 9 g of HMB per day for an average 60 kilograms body weight in humans. These results delineate neuroprotective functions of HMB and suggest that this widely used supplement may be repurposed for AD. There have been previous studies that indicated that a family of proteins, known as neurotrophic factors, are largely lowered in the brains of people that have Alzheimer's disease. These proteins have also been found to help in survival and function of neurons, which are cells that receive and send messages from the body to the brain and vice versa. All of that being said, what is the best supplement that contains HMB that can help with your fitness goals as well as give you a good amount of the supplement? Transparent Labs Strength Series Creatine HMB. Code GENERAN10 for 10% off. Transparent Labs Strength Series Creatine HMB. Transparent Labs Strength Series Creatine HMB. Transparent Labs Creatine HMB is clean and effective for tackling all of your strength building needs. With no artificial additives or preservatives, what you get is pure creatine with added HMB for better strength. Thank you for taking time to learn about this new discovery. Please like and subscribe, and come back to check out more medical discoveries.